Hello, and welcome to Cognitum, a show dedicated to exploring the present and future of science and technology. I'm your host, Iosif Gershteyn. Joining us today is Dr. Maxim Frank-Kamenetskii, Professor of Biomedical Engineering at Boston University and the author of Unraveling DNA, the Most Important Molecule. Hello, Maxim. So tell us, given your discovery of the importance of van der Waals forces for the stability of the double helix, what practical applications does this discovery have?
For most of application of DNA melting phenomena, which are many. In principle, this separation strand we talk about is enormously important phenomenon for application. For instance, it's suffice to mention polymerase chain reaction, DNA polymerase chain reaction, which is based on melting. But for majority of this practical application, the distinction between base pairing and base taking is not important because the stability determines by their combination anyway. So it is not that important to know which is dominant contribution. We found that it is stacking interaction, but it is not that practically important. But it is very important for future development of new types of life. It is not, it sounds like science fiction, but people now in many labs are trying to develop new type of life. All life we know is based on the DNA double helix, on this specific chemistry, DNA, and all genetic material for all living organisms is DNA double helix. And scientists started challenging this nature choice and try to develop artificial genetic molecule different from DNA. It may be RNA, or it may be not RNA, not DNA, some different chemistry and chemical structure. And for these attempts, it is very important to know which forces determines the stability of DNA and possible replacement of DNA. So in this case, it is very important to know how to design stable biological molecule, not DNA. But
so understanding the physics is going to be crucial.
Absolutely crucial, right?
For artificial nucleotides or non-ACTG
yes, because
nucleotides.
people already doing this. People already introduce into DNA non-canonical basis, and for understanding stability of the structure, it's very important to know, to design this, very important to know how to which interaction are crucial for the stability of.
And there is already work being done on DNA not using ACTG?
Yes.
And has that ever produced any proteins?
Yes, people already do this. It is extremely difficult, complicated experiments, and they introduced non-canonical bases, and they introduced non-canonical amino acids, which will be incorporated into proteins based on this non-canonical basis.
So we're opening up two whole new alphabets.
Absolutely.
And
it is what I myself work with artificial nucleic acid, but not in this context. But it is not my research. But people do this. People do this in different labs.
Now, if you had a license to speculate on where this could lead,
eventually, I'm sure it will lead to absolutely artificial life which radically different from life we know, life which we could expect to find on exoplanets, if we could ever reach them.
And what could we do with completely synthetic life?
Well, it is totally different ballgame. It opens up enormous possibilities.
And what of those possibilities would excite you most?
Well, I never thought about that far, frankly. But it is totally different. You cannot imagine what can be done with this. It is like it is artificial life.
Well, coming back to the life we know, the ACTG base pairs, we do have certain new technologies such as CRISPR that can alter our gene code now, right? And quite recently, a Professor He in China altered I believe the germline of some babies that were actually born with CRISPR for a CCR5 mutation to make them immune to HIV. I believe there was a great furor about this. What are your opinions about it?
First of all, we still don't know whether it is all true. We still don't know. Also, I think general attitude of scientific community is that it was probably all true, that he did it, and he is absolutely condemned by doing this. But we still don't have solid proof. Parents never appeared in any public space. These babies, nobody studied these babies. So it is all, we base this on the what this Chinese scientists did. And most of people mostly trust him because he is definitely, he could do this. He could do this. He has expertise to do this, proven because he got his PhD in the United States. He did postdoc in Stanford. So he is absolutely capable, as many other people in hundreds and hundreds of labs over the world can do this. He didn't do anything scientifically new. It is such things routinely is done with not with humans, of course, but with all other kind of other organisms, and he just applied it to humans, which was totally unethical and absolutely unwarranted. And I joined the chorus of all community which condemned him. I don't know where he is now. Nobody, I think, I haven't heard for a while anything about him. I look from time to time on internet trying to find new, but nothing new appeared recently about this. It was just very recently, just a letter signed by Chinese scientists condemning him, and if anything, it can damage in significant extent the prospect of real necessary advancement of this new technology, fantastic new technology in medicine, because it raises suspicion of public and all kind of government agency that scientists must be watched, must be under control, otherwise they can go wild, and it is totally unacceptable.
But the ethical questions aside, isn't this what CRISPR is moving towards? Isn't this what we're moving towards with genetic engineering. I mean, controlling the progression of our own gene code and,
yes,
and
of
how
course.
to change.
Of course, it is absolutely true, and it is inevitable. But it must be done with great caution, and it must be done without cutting edges. And it is very, very long way to apply this to especially there are two different things. One thing is to make genetically modified cells in somatic cells, incorporate them back to the organism for them to fight, for instance, with cancer and so forth. That's one thing. It is much easier. Of course, it also must be done with all kind of tests and precautions, but it is easier. But it is totally different situation when you genetically modify germ cell. And
germ cells are the basic cells from which an embryo grows.
Yes, this zygote you make from which the entire organism is supposed to.
So it's in the nucleotide of every cell.
Yes, and it will be in every cell. And here everything is much harder, because then it is essentially there's no way back. You already modified, which you changed the genome of baby and of person which will grow from this cell, and you don't know what will happen, and it is very hard to predict. And the this absolutely was unnecessary, because one thing, if you can expect this to be under supervision, under all kind of controls and all kind of precautions, you can imagine this, and probably it will happen soon. And some genetic disease which is absolutely lethal, and we know that if we do not fix this genetic defect, the baby will die very early, inevitably, 100%. We know this, and we get permission to fix it, and then at least this baby, maybe late in life, it will because of this the person will have more will be more prone to cancer or something, but at least the person will survive and not die early from this disease.
So weighing the relative risks of the intervention needs to be at the
on this stage it must be only in case the genetic defect is just lethal, and lethal early, not late in life, but early in life. And still it is questionable, but it is absolutely wrong what this Chinese professor did, to do it just to which are not necessary. AIDS is not that great threat, and that, yes, father of these babies was infected, HIV positive, but it doesn't mean that they will get HIV, and HIV is not that lethal any longer, and so forth and so forth. So it was absolutely and actually it is ironic, one of these there are twin.
Yes.
One of them it was heterozygous. So I think was girls. She still carries this non-mutant
CCR5.
CCR. Yeah. So she is prone to HIV as any normal human being. So what he did, he just put this go to all kind of negative possibility of negative side effects without any benefits. It is just so much terrible violation of a medical profession, unimaginable.
And so, when we move further in the science of genetic engineering, how do you see it happening? How do you see us integrating these technologies to increase or maximize human life and potential?
Well, in my opinion, with respect of this CRISPR revolution has many aspects. There are aspects which are absolutely clear with all kind of genetically modified crops and so forth. It is just much better, much more efficient way of do genetic engineering than it was before, and it is being done in huge scale now, and it will increase. I'm sure will improve all kind of agricultural application of.
But in terms of
genetic.
improving our own gene code.
In terms of human, I think it is. I'm not that optimistic that it will progress fast, because how possibly you can test for sure that the side effects are avoided. So again, only if you wait the inevitable death at the young age of the baby, and against this you can argue that you should do this, but otherwise you don't know whether if you want to improve something not dramatically lethal, then it always raises questions: what are benefits, whether benefits overweight the possible side effects. And we will not know about side effects until many people will go through the full life cycle, and it is like gerontology. Gerontology is very strange science. When you do say, you can claim that you do this, you will live very long with. How can you test this? You test this, you have to make clinical trial, and clinical trial must show that yes, people live, instead of 90 years, they now live 110 years. But how long this clinical
the
trial?
clinical trial has to last for 110 years. Yeah.
So it is very hard to do such things, and there are so many charlatans always. This fact always invites charlatans to surface, who will say, I sure you will live forever, but what is the proof?
Well, we always have to rely on a certain amount of faith whenever we take a new step. And when it comes to genetic engineering, we must have certain faith in our ability to predict the effect of changing the genetic code. Where are we right now in terms of predicting the effects of genetic alteration in humans?
No, it is the prediction is easy. If you change some gene, if you replace broken gene with good gene, everything will be fine with respect of this particular disease you are fighting. But with respect of side effects,
so the second order effects and the side effects are going to be unknown until we have a better predictive model, or potentially that predictive model.
Yeah, of course you can check this, you can check all kind of mutations, blah 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 blah. But the final analysis, you need to do this and check that nothing happens down the road, and it is very hard because the the bar is very high when we deal with humans. That's the reason.
Coming back to the example of Professor Hayes' CCR5 mutations on the twins, from what I read, he got permission from the parents to modify that embryo.
Yes.
So he did get informed consent to the extent that the informed consent has an informed aspect.
It was completely screwed up. As far as I know, what I read about this, it was completely screwed up because he himself got this permission from them, which is absolutely unacceptable. It is, according to all regulations, it must be a peer who is not involved in this work, but who is knowledgeable about this kind of things, who should talk to parents and get their consent on these experiments. Because if you are interested in convincing them yourself, how could we possibly be impartial?
Okay, I understand. So
this is totally unacceptable. It is absolutely wrong.
So the conflict of interest invalidated
terrible
the informed
conflict of
consent,
interest.
and therefore, in this case, the genetic alterations of the CCR5 were unethical.
It is totally unethical. You always can, especially if you talk with simple people who don't understand that you are a doctor and you know everything. You can talk to them to anything. It is just impossible. It is just absolutely unacceptable what he did. Totally.
So you believe that human genome editing is going to progress very, very slowly and will primarily be focused on monogenic diseases that are lethal?
Yes, simple monogenic diseases which has a simple like cystic fibrosis, blah blah blah. This kind of hemophilia thing where you can just fix one particular gene or even one particular mutation and so forth and so forth. And still, it will be very difficult to convince that it is still necessary, taking into account possible side effects. And so it is.
What's the time frame that you see this evolving in, just for the monogenic diseases that are lethal?
I don't know.
No.
I don't know. I cannot say. It is difficult to predict. It is very difficult to predict.
And the genetic engineering that we're seeing right now in laboratories, how is that affecting our ability to understand the human organism and to understand human physiology and biology?
You mean CRISPR?
CRISPR specifically, but
of course,
genetic engineering approaches
because
in general.
it makes tremendous impact because it allows to much easier to do things which before, for instance, knockout genes. It is technology, genetic technology, which allowed to eliminate specific genes. And it was very, very complicated technology. It existed, but extremely complicated. Of course, it mostly was done on mice and so forth, not on human. But most experiments which affect our understanding human, of course, are done on model organisms. And here, this technology has enormous impact because it is much, much easier to do all this kind of thing like knockout genes, replace one gene with another. It is just really easy. And
could you give a quick example of what a knockout gene and what a replacement gene is?
No, it is just it is the same. Knockout gene it means that it is just specific genetic technique to genetic engineering technique to eliminate the gene. And replacement is just replace it with other genes. So you can just remove the gene from genome, and you can replace, say, gene with mutation with normal gene, or gene normal with mutated gene. All this is much, much easier. Essentially, what is the difference between like old-fashioned genetic engineering, which appeared in 1970s, and this gene editing technology. The difference is that genetic engineering was that you take DNA from organism. You want to do something with genome. You take it out of the organism. You manipulate with it, and then you put it back. Here you don't need to do this. You do it in vivo. You do it in the living cell. You do genetic manipulation within living cell, not taking DNA and manipulating it. It is huge advance, huge advance. And this fantastic approaches, like when people this gene drive, it's called gene drive, when you change, you introduce this CRISPR in such a way that the organisms, say mosquitoes, they start carrying this in principle gene gene which is infertility gene. It spread through the population. How infertility gene can be spread in the population?
Horizontal gene transfer?
Yes, but infertility, it's horizontal gene transfer, exactly. But infertility, how you can transfer infertility?
Well, not by offspring.
Yeah, exactly. It looks like impossible. It is easy to do with CRISPR technology. And as a result of this, now we have this genetically modified mosquito, say malaria mosquito. If you put them into the ecosystem, all malaria mosquito will disappear, will die.
Yeah, and it's not going to be done through their offspring. It's going to be done through horizontal gene transfer. And could you explain succinctly what horizontal gene transfer is and why it's so important?
Yeah, because you have your well, it's a little bit technical. When you have normal Mendelian heredity, then you have when you introduce some gene and you get heterozygous, and then this gene will it will be difficult to spread, especially it is an infertility gene. When you get it in CRISPR, then you put it into the you modify the male mosquito, and this male mosquito then it gets offspring, and it is heterozygous, but within zygote it becomes homozygous. Mm-hmm. So this CRISPR allows to transfer this gene you want to spread, transfer it to the to mother chromosome. So it comes from father chromosome to offspring, and mother chromosome is normal, but within zygote it is transferred from father chromosome to mother chromosome. It become homozygous.
Mm-hmm. So,
and this infertility gene spread through population, and population die because they all infertile.
So in the zygote, the CRISPR modified male, the paternal germline actually modifies the female germline.
Exactly.
And therefore you can have hereditary infertility.
Yeah, it violates all Mendelian laws, and it is totally different genetics. And now the question only is whether we are, look, we still don't do this, although everything is ready, because we are concerned as society that although people die from malaria in Northern Africa, but we still do not introduce it into ecosystem because we are afraid that this horizontal gene transfer may happen to different species of mosquitoes, which are not malaria mosquitoes. And now all mosquitoes will start dying.
Yeah, and we just don't know
we
what
don't
kind
know
of
what
second
kind
order
of
and third order
effect
effects
it will
we could
produce
see.
on ecosystem. Yeah, mosquitoes part of ecosystem.
Yeah.
So that's why we still there are some. The same is for mosquito, from Zika virus, from West Nile virus. It is all possible to eliminate all these diseases. And there are some discussion in different communities, local communities, whether to do it locally. It is very, very complicated issue again, but it is now possible. It is just scientifically, it is absolutely proven.
Our possibilities exceed our understanding.
Yes.
Thank you so much for joining us today, Maxim. It was a pleasure. We hope you join us again next time as we explore the frontiers of science and technology with the thinkers creating the future. Good night. Farewell.